Dear viewers, this video is part 2 in the 4-part series on cancer cell biology in the Medicine for Anyone YouTube channel. Cancer as an organ that ignores its niche. The fundamental cellular defects that create a malignant neoplasm act at the cellular level, and some of these are cell autonomous. However, that is not the entire story. Cancers consist of both the malignant cells as well as other cells, blood vessels, extracellular matrix, and signaling and other molecules in the cancer microenvironment. They behave as organs that have lost their specialized function and stopped responding to signals that would limit their growth in tightly regulated normal tissue homeostasis. Most human cancers usually become clinically detectable when a primary mass is approximately 1 centimeter in diameter. Such a mass consists of about 10 power 9 cells. Often, patients present with tumors that are approximately 10 power 10 cells. A lethal tumor burden is usually about 10 power 12 to 10 power 13 cells, although it varies by type of cancer and where the primary tumor and metastasis are located. If all malignant cells were dividing without any cell death at the time of diagnosis, most patients would reach a lethal tumor burden in a very short time. So, dear viewers, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions, available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Not every daughter cell produced by a cell division is actively dividing. In addition, the overall growth rate of a tumor depends on differences between growth rates of different cells within the tumor and rate of cell losses. The growth fraction of a tumor, that is, percentage of cells which are actively dividing, declines with time, largely due to factors in the microenvironment. The growth fraction of the first malignant cell is 100%, and by the time a patient presents for medical care, the growth fraction is estimated to be less than 10%. This fraction varies between different types of cancers and even different cancers of the same type in different individuals. This fraction is often similar to the growth fraction of normal bone marrow and normal intestinal epithelium, the most highly proliferative normal tissues in the body, a fact that may explain the dose limiting toxicities to these tissues of agents that target dividing cells. The implication of this data is that the tumor is slowing its own growth over time. How does it do this? The tumor cells have multiple genetic lesions that tend to promote proliferation. Yet, by the time the tumor is clinically detectable, its capacity for proliferation has declined. Better understanding of how a tumor slows its own growth would provide important clues for better cancer treatment. A number of factors, including those in the tumor microenvironment, are known to contribute to the decreased proliferation of tumor cells over time in vivo. Some cells are hypoxemic and have inadequate supply of nutrients and energy. Some have sustained too much genetic damage to complete the cell cycle but have lost the capacity to undergo apoptosis and therefore survive but do not proliferate. However, an important subset is not actively dividing but retains the capacity to divide and can start dividing again under certain conditions, such as when the tumor mass is reduced by treatments leading to improved conditions in the tumor microenvironment, favorable for cell proliferation. Just as the bone marrow increases its rate of proliferation in response to bone marrow damaging agents. The tumor also seems to sense when tumor cell numbers have been reduced and can respond by increasing growth rate. However, the critical difference is that the marrow stops growing when it has reached its production goals, whereas tumors do not. The ultimate structure and organization of an organ are based on a number of factors including growth, migration, elimination, and death of various cells, communication between cells to establish the correct architecture, competition between cells, and the composition of extracellular matrix that is produced. In addition to normal cells stopping proliferation in an organ when that is appropriate, normal tissues have various mechanisms for eliminating cells both in the process of development as well as ongoing homeostasis of an organ. These include mechanical processes based on a number of factors including cell size, shape, and topology between cells that can determine cell fate, as well as an active process of cell extrusion, which plays a major role in the elimination of both cells that are no longer needed by the organ and hence cells that are damaged and potentially dangerous, such as those with mutations that might be precursors for malignancy. The process of cell extrusion may depend on cell cycle arrest in the S phase. Aberrations in this process may contribute to the metastatic process. A variety of processes, including major alterations in cell cycle control, apoptosis, and other mechanisms of cell death, and uncontrolled cell signaling, all contribute to defects in appropriate cell extrusion, contributing to the development of cancer. Additional tumor cell vulnerabilities are likely to be detected when we learn more about how normal cells respond to stop signals from their environment and why and how tumor cells and tissues fail to heed such signals. This figure shows the cell cycle. The normal cell mostly is in interphase, that is between mitosis and the next mitosis, G1 phase. In preparation for mitosis, the cell duplicates its DNA, that is the S phase. The cell checks itself for finally entering the mitosis, that is the G2 phase. Mitosis follows. When mitosis is completed, two daughter cells enter the ordinary growth phase, G1 phase. The checkpoints are shown as red cross sticks. Cell cycle checkpoints. See figure 1.1 cell cycle. The cell division cycle consists of four phases. G1 growth and preparation for DNA synthesis. S DNA synthesis. G2 preparation to divide. And M mitosis, that is cell division. Cells can also exit the cell cycle and be quiescent, that is G0 phase. Progression of a cell through the cell cycle is tightly regulated at a number of checkpoints, especially at the G1S boundary, the G2M boundary, and during M, that is spindle checkpoint, by an array of genes that are targeted by specific genetic alterations in cancer. Critical proteins in these control processes that are frequently mutated or otherwise inactivated in cancers are called tumor suppressor genes. Examples include RB protein, PRB, and P53. Role of PRB. Note, the retinoblastoma protein, PRB, is an inhibitor of cell cycle progression from the G1 to the S phase of the cell cycle. This protein acts as a tumor suppressor, which means that it regulates cell growth and keeps cells from dividing too fast or in an uncontrolled way. Under certain conditions, PRB stops other proteins from triggering DNA replication. Germline mutations in the RB1 gene, that is in chromosome 13, result in retinoblastomas, a rare form of childhood cancer, and also higher risk of osteosarcomas and other types of cancer. Germline, the DNA in germ cells, egg and sperm cells that join to form an embryo. Germline DNA is the source of DNA for all other cells in the body. In the first phase, G1, preparations are made to replicate the genetic material. The cell stops before entering the DNA synthesis phase or S phase to take inventory. Are we ready to replicate our DNA? Is the DNA repair machinery in place to fix any mutations that are detected? Are the DNA replicating enzymes available? Is there an adequate supply of nucleotides? Is there sufficient energy to proceed? The retinoblastoma protein RB plays a central role in placing a brake on the process until the cell is ready. When the cell determines that it is prepared to move ahead, sequential activation of cyclin-dependent kinases, that is CDKs, results in the inactivation of the brake RB by phosphorylation. Phosphorylated RB releases the S phase regulating transcription factor E2F-DP1 and genes required for S phase progression are expressed. If the cell determines that it is unready to move ahead with DNA replication, a number of inhibitors are capable of blocking the action of the CDKs, including P21, CYP2, BAR, WAF1, P16, INK4A, and P27, KIP1. Ready for cell division leads to cyclin-dependent kinases release and releases the brake of protein RB, that is retinoblastoma protein. PRB is phosphorylated, that is activated. Then the release of S-phase transcription factor E2F-DP1. Then the genes required for S-phase progression activated, and then cells proceed for cell division. If the cells are unready for cell division, cyclin-dependent kinases activity is blocked by P21 CYP2 WEF1 or P16 INK4A or P27 H1P1. Nearly every cancer has one or more defects in the G1 checkpoint that permit progression to S phase, despite abnormalities in DNA repair machinery or other deficiencies that would affect normal DNA synthesis. At the end of the G2 phase and prior to the M phase, after the cell has exactly duplicated its DNA content, the second inventory is taken at the G2 checkpoint. Have all of the chromosomes been fully duplicated? Were all segments of DNA copied only once? Has all damaged DNA been repaired? Do we have the right number of chromosomes and the right amount of DNA? If so, the cell proceeds to prepare for division by synthesizing mitotic spindle and other proteins needed to produce two daughter cells. So, dear viewers, this is the end of part 2 in the 4-part series on cancer cell biology in the Medicine for Anyone in the YouTube channel. Thank you for watching the video.